microphone so that the only person we hear is the speaker, who is Denis McCarthy from the psychology department. So Denis wants me to point out that he is a clinical psychologist. He's not a pharmacologist or any of the other ologists who are associated with alcohol and addiction research. And so he's interested in the effects of and why people do it, and not various receptors that one might find located in different places in the brain. So his academic career was he was an undergraduate at Notre Dame, and then he got his Ph.D. at University of Kentucky, and then did his postdoc and was a research scientist at UC San Diego before coming here, and this is where he spent his academic career. Let's please welcome Denis. I think it'll be a really interesting talk.
Hi, thanks. Microphone okay? Yeah. So yeah, I'm an alcohol researcher, and I came here from UC San Diego. And when I was telling people that I was accepting a position at Mizzou, and I'm in San Diego, everyone's like, "Why are you doing that?" But if they were alcohol researchers, they all understood, because Mizzou has been on the map as an alcohol research hub for a long time. And especially in the psychology department, we have about 10 or 12 people that do addiction and alcohol-related research, and it was a great first job. I've been here ever since. It's also a much better place to start a family, no offense to Southern California. But the title of my talk is What Happens When You Drink and Why You Should Care. I know we have time for questions at the end, but I like having questions in the middle, so I'd be glad to kind of derail the talk. I'm not that attached to my slides, so if you want to ask and go more in depth on one thing, with the caveat that I'm not a biologist or pharmacologist, so I may not know a lot of detail about the biology, but the research on alcohol in general I know a lot about. I love being an alcohol researcher. I do other drugs too a little bit, but I love being an alcohol researcher because it affects everybody's life, right? My friends that do schizophrenia research, they're addressing an important topic in psychology, but less than 1% of the country has schizophrenia. A lot of people go through their whole life without dealing with schizophrenia. Nobody goes through life, in the US at least, without dealing with alcohol. Even if they are themselves an abstainer, alcohol has affected their life. Even if it's just someone being a jerk to them because they're drinking, or someone in their family having an alcohol problem, or things along those lines. So it's something that everyone has opinions about, everyone is affected by in some way. The problem with being an alcohol researcher is that everyone is not, affected by it in some way, and everyone has opinions about it. So the very common thing I hear from people is, why do we need alcohol researchers? Millions of people drink alcohol every day. Don't we already know everything there is to know? Or something along the lines of, I drink a lot of alcohol, I could probably take your place, right? And there are some frustrations about that. And so one of the things my talk is about is, well, another version of that is friends of mine say, well, wait, what do you do all day? I said, well, my research is I bring people into my lab. I get them pretty drunk, and then I have them do stuff. And you get a couple of reactions when you tell people that's what you do. They say, well, A, I'd like to sign up for that. Especially because we pay them about $100 to $150 for doing it. Two, they say, well, why do you need to do that? Like, I do that every day. Why? What's the value of studying that, right? And so my talk is kind of geared toward both of those things. But one of the things that comes up in alcohol research is, why is there such separation between alcohol research and some other areas of research, or some other addiction research? And I always argue that alcohol is a very unique drug for both chemical and societal reasons. It's a unique substance in our lives, right? One of the reasons is kind of obvious. It's the only legal impairing drug. It's not the only legal addictive drug, right? And I'm not saying that that's true about that there aren't prescription medications, right, that are addictive. But alcohol is legal for adults to use pretty much however much they want. There's not a legal restriction on how much you can purchase, how much you can drink, right? And a majority of people in the US at least drink some, right? Close to 80% of US people have at least drank alcohol in their lifetime, right? And a majority, over 50%, drink alcohol on a somewhat regular basis, at least once or twice a year, right? So that changes how we think about research on alcohol. we don't think about alcohol research in quite the same way that we think about cocaine research, right? When we're doing alcohol research, everything we do is predicated on the idea that some people will be exposed to this chemical, right? When we're doing cocaine research, we're thinking, don't do cocaine, right? And we're thinking, how can we get people to either not use cocaine or not get addicted to cocaine or get them off cocaine once they are? But for alcohol, we think people are going to do this. The law allows them to do this. So how can we help them do this in a way that reduces harm, whether that's physical harm, behavioral harm, cognitive harm, whatever, right? And so a lot of that research leads us down the roads of what happens when you drink and how do those things differ across individuals. Let me see if no, it's not working. How do those effects differ across individuals? And then how do those differences impact risk? Right? Are some people extra sensitive to alcohol? Are some people less sensitive to alcohol? Do some people experience different effects when they drink than other people? And how do those differences explain the kinds of problems they have? Again, whether they're biological problems, health problems, or whether they're behavioral problems. Another thing that's different about alcohol is that it's a dirty drug. We call it a dirty drug. And when I talk to pharmacologists or neuroscientists, a lot of times they say, well, I haven't really thought about this because alcohol is not my drug, but where are the alcohol receptors? And my answer is there are no alcohol receptors, right? There are cannabinoid receptors, there are nicotinic receptors, right? There are certainly opiate receptors, right? And a lot of those, the drugs are drugs of abuse because they mimic something that's already going on in our body that we've already developed receptors for, right? Alcohol is not like that. Alcohol is probably older than a lot of these other drugs. It's always been around because it's produced by fermentation. And all biological animals, to some extent, have the chance of running across alcohol in their environment, right? So from fruit flies to elephants, right, you could be exposed to alcohol. It's a toxin. Your body has to have a solution for breaking it down, right? But because there's no receptors, it has significant effects beyond the brain. In general, alcohol distributes pretty evenly in total body water, right? So most of your body other than bones and fat. And it has significant effects on almost every type of cell we have. And so then alcohol research has to say, not just how does alcohol affect the brain, how does it affect reward circuitry and produce addiction, but also how does it affect your liver, how does it affect your pancreas, how does it affect your esophagus in a way that we don't necessarily do with cannabinoids, for example. Okay? Yeah. So what I'm going to talk about today is really basic alcohol metabolism, right? Just the idea of what happens when you drink, how your body breaks it down in the main biological pathway, right? Then how does alcohol affect health? That's a really important issue that's been going on. There's been a lot of news about it. That's something that I'd totally be willing to be sidetracked about, but I'll kind of hit the highlights. And then I'll talk a little bit about my kind of research. I won't talk too much about my personal research, but the science of intoxication, like why do we study people who are intoxicated in the lab? What do we learn from that, and what are the kind of societal benefits from understanding that process better? Okay. And just briefly, so I'm going to talk about that in terms of addiction to alcohol, in terms of judgment, and then something called acute tolerance, which I think is, it's not unique to alcohol, but the research on it is relatively unique to alcohol. It's something that a lot of people don't know about, and I think is a very important contribution to why alcohol creates risky behaviors. Okay, so basics of alcohol metabolism. When you first drink, it goes into your stomach. I always say that your body has a totally different plan than you do, right? So like when you're trying to lose weight and you cut down on how much food you have, your body stubbornly decides to slow down your metabolism, right? Similarly, when people drink alcohol, a lot of times they're trying to get an effect from alcohol. And your body does not have that plan. Your body's plan is to eliminate this toxin from the system, right? Again, this is a toxin that you could naturally come across if you were eating fermented fruit, right? And so your body is treating it as a toxin that you've done. When it's sitting in your stomach, it doesn't really do anything. It's not relevant pharmacologically to your psychological effects until it leaves the stomach and enters your bloodstream. So it's sitting in your stomach for a while. You actually have enzymes in your stomach that break down alcohol. So the longer it sits in your stomach, the more alcohol will get broken down before it ever gets to your bloodstream. So if you drink a small enough amount of alcohol, right, what you might come across if you actually ate a fermented piece of fruit, and you had a pretty full stomach, you might feel no effects because it might all get broken down in your stomach and never actually get to your bloodstream. What we do as humans, and we've done for more than 10,000 years, is overload that system, right? We ferment stuff ourselves, and then we swamp the system with way too much alcohol for our stomach to handle. Then it dumps it into your digestive tract. Ideally, it shunts it straight to the liver. Again, if it's a small enough amount, it shunts it to the liver. That's where the majority of the alcohol dehydrogenase enzymes are, and they would break down alcohol, and you would, again, feel no effects. But you've overwhelmed it. It has to go somewhere. It dumps it in your small intestine. Then it starts getting absorbed into your bloodstream. It's kind of a storage place for it while the liver continues to process it throughout the next couple of hours, right? So the basics of it, it's not a one-step process, it's a two-step process, and this is the main oxidative pathway. There are secondary oxidative pathways and other pathways. This is the main one, that alcohol dehydrogenase first breaks down alcohol into acetaldehyde, right? What's interesting is that both alcohol and acetaldehyde are toxins, and both of them have psychological effects. So some of the things that we think about as being the effect of alcohol are probably actually the effect of acetaldehyde, right? How come we don't know a better, how come we can't say, oh, this is the effect of alcohol, this is the effect of acetaldehyde? We can test really well how much alcohol is in your system. Alcohol distributes pretty evenly in your total body water. That's why taking a sample of your blood or even your breath will give us a good representation of how much alcohol is in your bloodstream, right? And then how much alcohol is in your brain, which is what we usually really care about. Acetaldehyde doesn't do that. So we can't take a sample of your blood and say, this is how much acetaldehyde you have. So when we give someone alcohol, we can be monitoring their blood alcohol concentration on a continuous basis. We can't monitor their acetaldehyde level, right? You can do it in animal research, but it's too invasive to do for humans. I'm not even totally sure of the method, right? So some of the effects we have from alcohol, some of the effects we have from acetaldehyde. I'll talk about a couple of ways we can make some inferences about the difference between the two, but some of it remains a question, right? Then aldehyde dehydrogenase is the second enzyme that's largely responsible for breaking down acetaldehyde. It breaks it down into acetate, which is harmless. That can then be passed from your body, and it's not toxic, right? So I've already said a lot of this, right? Breaks down into acetaldehyde, then an acetate. Both of them have distinct cognitive, behavioral, and kind of iatrogenic effects on tissues. So I've talked about alcohol as a toxin that your body's trying to get rid of, right? What are some of the basic things we know about what alcohol does as a toxin to your body, right? We know that alcohol creates increased risk for a large number of health problems, right? A lot of forms of cancer, incidences that go way up when people are drinking. You know, and when I say way up, maybe doubles or triples, right? But some of these are relatively low base rate forms of cancer. We're getting really good at telling even what type of cancers are caused by alcohol. But cancers of the mouth, cancers of the stomach, esophageal, colorectal, all go up. That's because alcohol is hitting those tissues when you consume it, right? Obviously the liver, right? And that's something that I think everyone thinks about when they think about alcohol negative health effects, right? Everyone knows about cirrhosis. Most people know about alcohol-induced hepatitis, fatty liver, right? Those are things that are a consequence of significant alcohol consumption, right? Alcohol also causes cardiovascular problems, right? It causes problems with the heart, cardiomyopathy, arrhythmia. It can also cause some forms of stroke, and I'll talk a little bit more about that, right? So this is pretty bad. Oh, and pancreatitis, right? So again, it distributes pretty evenly through your body. So when it touches the pancreas, it can have negative effects and cause pancreatitis. This is pretty bad, right? And alcohol does a lot of bad stuff. Why do we keep hearing about the health benefits of alcohol, right? And we do keep hearing about it. The reason we keep hearing about it is because when we do epidemiologic studies, this J-shaped curve keeps showing up, right? The J-shaped curve is the idea that at zero consumption, right, all the way at the left side of the x-axis, right, people are a little bit less healthy than at 1 or 2 drinks per day, right? So when you go from full abstention to 1 or 2 drinks per day, there's a slight improvement in health, again, from epidemiologic data, right? Then as the number of drinks go up, this actually is not a good, I should have got a better version of the J-shaped curve, because really it probably now we think it starts going up right after 2, right? So around 2 to 3, it starts going up, and eventually if you're drinking 4 or 5 drinks per day, you're worse off than abstainers in terms of general all-cause mortality, right? So this, again, epidemiologic research, the y-axis can either be all-cause mortality or it could be cardiovascular problems, right? So again, abstainers versus 1 or 2 drinks per day, 1 or 2 drinks per day tend to show slight improvement in cardiovascular problems and all-cause mortality, but it goes right back up and gets much worse pretty quickly, right? This is the basis for people thinking that there's a health benefit of moderate alcohol use. Yes?
So is there a difference in the eating of people that have 1 or 2 drinks a day?
Yeah, so that's a very good question. This is epidemiologic research, and I'll talk about the potential problems, but you're hitting on one, which is selection bias, right? People like this J-shaped curve. Scientists like it, right? Because scientists like shapes that keep appearing in nature, right? The J-shaped curve appears in nature a lot, not in nature, but in our study of different natural phenomena, right? The J-shaped curve is pretty common across a lot of other phenomena, so they're like, oh, just like these 3 other phenomena, J-shaped curve, right? We like logistic curves for the same reason, right? So we like things that's like, oh, this forms a kind of reasonable rule-based pattern that we're used to, right? But it also just keeps showing up when we do epidemiologic research, right? So given that it's a toxin, how could that possibly be true? Yes?
Why is it not wine?
Oh, good question. It's not wine because you think it's wine because of the wine industry, right? The wine industry has sold people, yeah. So this J-shaped curve shows up with the same structure when you look at any type of alcohol. The wine industry has done a pretty good job of convincing people that the tannins and antioxidants in red wine are what explains this, but this same curve shows up when we look at any type of alcohol, right? So my parents drink red wine every day, right? And they say this is why, right? You know, if my father was dying to drink a glass of bourbon every day, I would show this to him and then he would switch to that, but he likes red wine, so it's fine. But I think people wanted to believe that, right? But as far as we can tell, the process that produces this J-shaped curve from alcohol is not different for wine and other types of drugs. Okay, but there's a lot of caveats to that, and I'll get to that. Okay, so good question. So if alcohol is a toxin, how could it possibly have health benefits, right? Well, there's a couple mechanisms that, one, there's some good evidence for, one is a little bit more speculative, right? We know that alcohol has anticoagulant effects, right? So one of the ways it improves cardiovascular health is by reducing incidence of ischemic stroke, right? And we do know alcohol does do that, right? Is it enough to produce these health benefits? Not totally clear, right? Because, of course, if it's anticoagulant and it reduces ischemic stroke, it might increase the risk of hemorrhagic stroke, right? It's not a really strong anticoagulant, right? It's not something I would take instead of an anticoagulant, right? That's the reason we don't prescribe alcohol as an anticoagulant. But it does have a small anticoagulant effect, and that might, over time, reduce the risk of ischemic stroke, right? There's also some light evidence that it might improve arterial functioning. They think some lipid transfer might improve kind of the quality of arterial walls. I don't find that research compelling, but that is a suggested mechanism. Okay, so is this enough? Yeah.
Well, I thought of, not knowing the science of it, is it just that some people actually, relax enough in their day at an important point?
Yeah, so actually a whole other talk that I give is another interesting novel thing about alcohol is that people drink it for different reasons, right? Alcohol has both sedative and stimulant effects, right? And that's true of a lot of drugs. So if you think about any drug that you take, right, it has an opponent process function, right? Where the initial effect is X and then you have an opponent process Y later, right? Alcohol has an initial stimulating effect, right? That's why people drink alcohol when they're going to go out dancing. That's why they drink alcohol when they're going to go party, right? That's why they are probably drinking now before they go into the game, right? They're not drinking for the sedating. I want to be nice and relaxed in case Mizzou, blows it, right? That's not, right? But people do drink alcohol to relax at the end of the day, right? Alcohol does have sedating effects later, right? The stimulating effects are pretty short-lived, right? And that's why we talk about alcohol as a sedative, right? The interesting thing about alcohol that's different from other drugs, because again, they all have that opponent process. Both of those are motivationally relevant. As a psychologist, I get interested in this, right? People drink for both reasons. People don't use like crystal methamphetamine so that they can get a good night's sleep 3 days later, right? So, you know, it does, of course, let you do that, right? But that's not a motivation. And from a psychologist's perspective, having a different motive for the behavior changes the behavior, right? So drinking to party and drinking to relax are different behaviors that have different effects, right? And people do both. I think your question, though, about its effect on health goes back to the question, which I'm about to get to, about selection bias in epidemiologic studies, right? So in general, the reason we can't come up with a really good answer to this, people have said alcohol has this mild health benefit, right? But humans are the worst research subjects, right? They are terrible. Now, just to be clear, like some of my friends are humans, but they're really bad to deal with. So think about how we have to do an epidemiologic study, even if we do well-funded, well-conducted, longitudinal epidemiologic study on regular alcohol use and health benefits. That's going to require people remember accurately how much they drank, report accurately how much they drank. Don't really deviate from that, right? Because we know that if you get above 3 drinks a day, these health benefits go away, right? So there's a lot of potential problems of people telling us that they have 2 drinks a day and that's all they ever have and they never go above that, right? Who are those people? Are they people that do that, or is there something else about those people, right? So people are bad reporters, rememberers, and things like that. So even the best, well-done epidemiologic study is not going to provide us the kind of data we want to make a very firm conclusion on this. This is why, in general, right, the FDA has not changed their recommendations to tell people who don't drink that they should start drinking, right? Even though the J-shaped curve has been there for 20, 30 years now, a lot of people, some of my friends, are very convinced by it. We don't tell people, start drinking, it'll help you, right? 1, because we're not 100% confident in this. 2, because it doesn't take much to tip over into negative health benefits after 2 drinks a day, right? So those are the 2 reasons. Getting back to your question about, kind of who's in the study, who's not in the study, right? There's huge selection biases in who is an abstainer and who's not. The classic and biggest effect is probably the sick abstainer idea. People who have another illness are likely to abstain, right? That could make the abstainers look sicker. It has nothing to do with alcohol, right? Think about if you have a lifetime illness, right? So my sister has seizure disorder, and since she was young, she's always had to take medication for which alcohol is contraindicated, right? She still drinks. She shouldn't. Sorry if she's watching this on YouTube. But she shouldn't be drinking on the medication that she's on. She goes in the abstainer category, right? Now I use her as an example because seizure disorder doesn't, the way she has it certainly, doesn't really increase all-cause mortality for her, right? But think about sicknesses that do. And a lot of people abstain because they have an illness, right? Or they're drinking, they get sick, and then they stop drinking if the alcohol maybe even caused that illness. And then do they report accurately why they're not drinking, right? So the beginning, just to go back to the J-shaped curve, the beginning of that J-shaped curve, people all the way on this side of the x-axis in the 0 category is probably a pretty heterogeneous category, right? It's not people that are just deciding not to drink because they don't like alcohol. So we can't say that the difference between 0 on the y-axis and 1 and 2 is only alcohol. It could be a lot of things, right? A countervailing example is if religiosity is why you're abstaining, religiosity is related to improved health, right? So it should actually make this effect bigger, right? Because, well, no, it should be eliminating this effect, and it's not, right? So there's a lot of potential biases in epidemiologic data that reduces our confidence in this. Does that make sense to everybody? Okay. So when I say that to people, they always say, okay, why aren't you doing an experiment then, right? That's what scientists always do. If you have something that you really want to know the answer to, and you've collected lots of good epidemiologic data, lots of good data from other types of sources, mechanistic studies and things like that, The answer, if you really want to get at causal inferences, is an experimental design, right? Random assignment to abstention or not, get rid of these biases and things like that, right? Remember, one, that humans are the worst research subjects, right? So let's say, first of all, this has been done over the last several years. It's the first time ever that we kind of decided this was worth doing. That NIAAA, which is National Institute of Alcohol Abuse and Alcoholism, federal agency that funds a lot of my research, and it funds a lot of research on alcohol and health, but also alcohol and risk. And they decided the evidence is clear enough that it's worth the risk of doing an experiment, right? It was a huge $100 million study. They were running it for 10 years, and people would be randomly assigned to abstention or 1 to 2 drinks a day, depending on some things, right? This would give us a real answer to this question, okay? But just out of the gate, there are some problems with that, right? One problem is condition adherence, right? Humans are terrible research subjects. So imagine that you were assigned to whichever of those 2 conditions doesn't fit for you. Let's say you're an abstainer, and you're assigned, I'm sorry you're an abstainer, but you've been randomly assigned to drink 1 to 2 drinks a day. You're like, that seems like a lot of work, right? Now imagine you drink 1 to 2 drinks a day, and they say for the next 10 years, I'd like you to never drink anything, right? How good are you going to be at that adherence, right? That's always been a problem with this design, okay? And are you going to, we're paying you to be in the study, we're following you up regularly. How good are you going to be at reporting accurately whether you're adhering to the condition, right? Experiments are great, but here you're giving up control of adherence to the condition to the people on a daily basis. That's really hard. So that's always been a problem with this. The other problem is you have to exclude people if being in this study would put them at risk, right? So for example, people at risk for certain type of breast cancer that we know is exacerbated by alcohol, we can't include them, right? I mean, just ethically, you can't include them, right? People criticize the study because they excluded them. They said, you're biasing the study toward finding the J-shaped curve, right? Finding health benefits of alcohol. It's like, yeah, I get that, but on the other hand, you can't put people at risk in your experiment. Well, I'm sorry that I'm increasing your risk of cancer knowingly, but I really want to know the answer to this question, right? So that was hard, right? But they put this together. They made I think they made a lot of right calls about the exclusion criteria and how to conduct the study. But has anyone followed this? It's been all over the news, right? Does anyone know the mock study? Besides Carol, because she's cheating. No, so this was all over the news. It's funny that people, so you followed this a little bit? Well, no, that's a different study. So there's been a lot of studies in the news saying, we've looked at this and we don't support the J-shaped curve. But again, those are epidemiologic studies, and there's been a little bit of contradictory evidence. I think most of it has to do with how they treat sick abstainers, right? But no, this was a little bit different. This study, $100 million study, is supposed to go over 10 years. It was in year 3, I think. And records started to indicate that the funding agency had talked to the beverage industry about funding the study. We all knew that the beverage industry was funding the study. That was not a secret, right? They spent $60 to $80 million toward the study. They can donate to a foundation for NIH and earmark it for a study. They did that. I was actually fine with that. The problem was that meeting notes and other things came out that people from NIAAA were talking to the beverage industry saying, hey, you should fund this study. It'll probably be good for you, right? And that got people very concerned, right? I actually know a couple of people that were doing that. I understand why they did it because they totally believe in the J-shaped curve. So they didn't think of this as bias, but to just about everyone else that is agnostic about whether the J-shaped curve, how this would come out, they thought that was a real sign of bias. And so there was such a big uproar about it that this study was discontinued. So we will never get results of this study. It went for 3 years, but that's not long enough, right? So we won't do this, and we probably will never do this experiment, right? So we probably will never have this answer for sure, right? So that's one of the big things I wanted to talk about today because I think it's something that everyone always tells me, and we're Americans, right? Or maybe a lot of you are Americans, maybe not all, but as US citizens, our culture is such that I know a lot of people like, I heard that alcohol drinking is, so I've been doing a lot of it, right? We're the country of all-you-can-eat buffets, right? You go to Italy, they don't have all-you-can-eat buffets, even though Italians like to eat. So it's hard for us to hear a message of 2 is good, but 4 is not better, right? So I think this is something that we as a society and just nationally will struggle with whether this is right, and we will not get an answer. So I want you to be thinking about that. If you take anything away from this talk, it would be to think, yeah, I should be skeptical about when the news reports, so this is my thing, right? Whenever the news reports on a study, it's almost always wrong because they act like this is the conclusive thing, right? Everything you read about this will always be epidemiologic because we're probably not going to do this study. Even if we did redo this study, it would take 10 years, and I'm not convinced that 10 years is long enough, right? So any evidence you hear about the health benefits of alcohol or its negative effects are going to be based on data that are not conclusive, if that makes sense. Yes? Yes, I mean, this is something that a lot of countries are very interested in, right? Yes, are other countries doing this? Like, are any other countries going to fund it? Yes, but I don't think they would fund it on the scale of this, and I think after this experience, I think a lot of other countries, like the European Science Foundation, I'm not even sure what it's called, but people in Europe that fund research, they have a stricter attitude about some of these things, so I don't think that a beverage industry in Europe is going to fund this kind of stuff, and I don't think people would believe it if they did. So I'm not sure that the experiment will ever be done. Okay, so that's one thing I wanted to talk about. The next thing I want to talk about is drinking in the lab or the science of intoxication. What do we get out of doing studies where we get people drunk and seeing what happens, right? One of the main, so first let me just talk about some basics, right? When I actually read a paper recently where someone was doing a study very similar to one of mine, but they did it in the bar, and they actually had people responding to things, and in the conclusion they said, well, there's all these problems with this study because people were drinking however much they want. We could have brought them into the lab and got them drunk, but that's not really ethical. I was like, huh, it's funny. I do that all the time. But you have to be very careful about it, right? So, of course, they have to be legal drinking age, right? They have to also be non-abstainers, right? And part of this is an informed consent issue. I can't give someone a volume of alcohol that they have not voluntarily given themselves in the recent past, right? Some of that is I'm not wanting to add to their risk if they don't drink, right? But some of it is they can't, if they're like, I've never had more than 1 drink, but I'm ready to have 5, right? That's not a good informed consent process, right? Now, it's interesting because, of course, we give people drugs they've never had before in clinical trials, right? But in those cases, it's a risk-benefit ratio. We're trying to treat something, right? Here, we're just trying to learn something. So the risk-benefit ratio is such that we can't give someone a volume of alcohol that they have not voluntarily given themselves recently, right? Of course, so back in the old days, they just used to run men, right? Important progress that we don't do that anymore, right? Downside to that is, of course, you cannot give pregnant or nursing females alcohol, right? Even if they want you to, even if they are drinking on their own. They're like, I don't care about the negative effects of alcohol. I'm 3 months pregnant, but I'm drinking every day. I can't contribute to that, right? But the corollary of that is then I have to give people pregnancy tests, right? The corollary of that is sometimes they test positive in my lab, right? The corollary of that is they weren't expecting that. Right? Because they showed up to my lab to drink alcohol, right? So that doesn't usually go very well, but it's kind of part of the deal. We dose them by body weight and gender. What we really should do is dose them by total body water, right? So what we should do is we should measure their fat percentage, and we should give them, so a 150-pound person and 250-pound people, one of them has almost no body fat and the other has 15% body fat. They should get different doses, right? But it's stressful enough that I put people on a scale at all. And they're already annoyed enough about it. I can imagine if I brought out the calipers and tried to give them their body fat percentage. So I don't do that. We make an estimate of it. It's a little bit sloppy. It's not as precise scientifically as I would like, right? I give people alcohol orally, right? They self-administer it. which is great. It's an old technology that people have used for thousands of years. But my friends at different universities do intravenous infusion of alcohol. From a scientific perspective, much better, right? Very precise dosing, very clear control over how much they have, what their BAC is. You can even bring them up really quick, keep them there for a while, bring them down, bring them back up. You can do lots of cool stuff that I pretty much can't do, right? The downside is, remember we talked about how alcohol gets metabolized through your GI tract. You're bypassing that. You're just dropping it directly into their bloodstream, right? So from a psychologist perspective, I don't like IV because it has no external validity to the regular drinking experience that people have, with some exceptions of people that do that, right? It's a terrible idea, don't ever do that. And I had a friend in college that was convinced if you did that, that you could pass a breathalyzer. And that's not true. Just to be clear, the reason breathalyzers pick up the alcohol is because of alveoli transfer and carbon dioxide. It's not because the alcohol went in your mouth. Okay. I give people a bolus dose of alcohol, meaning I give them a lot of alcohol designed to get them up to about 0.1. They drink it in a relatively short period of time. We wait about 15, 20 minutes, and then we start doing things. They're kind of interesting to me for about 2 hours after that, right? That's not how people drink, right? So that's a problem, and it's an external validity problem with the design that I use. It has a lot of other benefits, but very few people say, I'm going to drink 5 shots right now, and then I'm just going to sit around, right? It's usually, so just to get ahead of myself, right? This is what the blood alcohol curve looks like in my lab, right? Where people start at 0, they go up to a certain level, then it starts gradually coming down based on their alcohol elimination rate, right? Yeah. Well, that depends, right? So in this case, it depends on dose, right? Because people move out. For what I do, it's about an hour, 45 minutes to an hour is peak, right? And then elimination rate is about 0.01 to 0.02 and change per hour, depending on the person, right? Which doesn't sound like a big variance, but it's actually double. So some people eliminate alcohol twice as fast as other people, right? But the absorption rate does vary, but I would say between 45 minutes to an hour in the dose that we give. Okay. But it also, so we give them a light breakfast, right? We actually give them Gino's pizza rolls, which my student came up with, I really liked, because we can actually titrate how many pizza rolls they get based on their weight, based on their expected caloric intake per day, right? So it's like, oh, you're a 7 pizza roll person. We have no complaints from the 21 to 25-year-olds about the pizza rolls. But you have to give them some amount of food, right? Otherwise, they'll absorb alcohol too quickly. We give them, we use Everclear and orange juice now, right? We used to do tonic. Tonic tastes horrible. It's good for placebos, though. So we give them Everclear, which is a pretty strong dose, even mixed with the orange juice that we give them. So that also makes a difference, the vehicle or how much is in the vehicle. So it's not that dilute is another thing. So that's why we wind up with 45 minutes to an hour. Okay. But I should mention that other labs do self-administration, right? Where you give people a dose and then you let them drink how much they want, right? That gets risky, of course, right? You can't let them drink as much as they want because that could be dangerous, right? But it also, from a scientific perspective, gets a little bit messy, right? Is probably the simplest way to put it. It's better because it's more externally valid because that's what people do. They administer alcohol based on how much they want, right? But, so there's lots of things about my lab and the environment that is not very externally valid. It's also in one of the buildings on campus, right? It's not the time of day, you know, it's relatively late morning, but it's not evening, right? And it's in a professional looking building with professional staff. That's not usually the environment that people drink in. So there's a lot of issues with that. Once they're done with my protocol, I have to keep them until they're relatively sober, right? So they have to be below 0.04. I like to get well below 0.04 before I let them go. But even so, we get them a taxi, and they sign an affidavit saying that they won't do any driving or work or leave their house for the rest of the day. Right? For whatever that's worth, right? People know that they could violate that affidavit, but We really strongly emphasize to them. Every once in a while I have someone, as soon as they're done, they're like, I want to leave, right? I usually meet with those people individually. I tell them I'll pay them more, right? Don't sign up for my study and then do this, but I do tell them I'll pay you more if you stay. I really, really don't want people to leave, and because we would have an issue if someone went and did something and got themselves hurt later that day, right? From a would I get in trouble standpoint, I'd have to document that I dotted all my I's. But also, I just don't want that to happen, right? Okay, and then last thing, there are different designs, right? You've probably heard about placebo. Alcohol is one of these drugs that we can do really good placebos with, right? I do placebo, I don't do a fully balanced placebo design, but I do placebo design. So people get almost no alcohol and are convinced that they had at least a couple of drinks, right? And as a psychologist, there's a lot of things I need to know from a placebo design. You can also do an anti-placebo. I haven't seen an anti-placebo design in about 15 years, but anti-placebo is to tell someone there's no alcohol in it, but give them alcohol. If you want to make the full inferences about pharmacology and expectancy, you need to have that condition, but that condition is very ethically risky. Right? Telling someone there's no alcohol in this and then getting them up to 0.1 is ethically risky. You really have to have an important reason to do that. I haven't seen anyone have a good enough reason in about 15 years. Okay, so I've already said a little bit about the blood alcohol curve, right? We're interested in people once they get a little bit before the A and then about halfway down the blood alcohol curve. That's when we do our assessments. Okay, so I'll come back to those ideas as we go. So we usually go till
10 more minutes?
10 more minutes? All right, I'll go quick on some things. So one of the things that we've learned from this type of research is that people vary a lot in how strongly alcohol affects them, both subjectively, physiologically, behaviorally, all the things that you think alcohol does. People vary a lot. Even at the same BAC, people will produce different effects. Some people are sensitive, some people are insensitive to alcohol. It's a general global phenomena. And overall, what we find is people that are insensitive to the effects of alcohol are at increased risk for alcohol use disorder. Right? It's a little bit surprising to think of it that way because also from a kind of reinforcement perspective as a psychologist, I think, well, if they get a big bang for their buck, that should be more addicting, right? But people that have to drink more alcohol in order to get the same effect as other people tend to be at increased risk for alcohol use disorder, right? There's probably some social conditioning things to that, right? Everyone tries to get to the same level of intoxication as their friends. If it takes more volume for you to do that, over the years, you're increased risk for developing a disorder, right? So there's a model that says we should really think of this as a unitary phenomena where low response is a thing that exists across physiological, subjective, impairments, judgment, things like that. There's a differentiator model that talks about the difference between stimulation and sedation, right? Because alcohol does have both effects, and that maybe risk is heightened for people who are sensitive to the stimulating effects of alcohol and insensitive to the sedating effects, right? And that you could see why that would be the most worrisome profile, right? So they get up really quick, they're ready to party, and they don't get tired as the drinking session goes on, right? So that's a more common thing. All this is complicated, though, by the fact that we're talking about their initial response to alcohol. When they're, as an organism, they're naive to alcohol. But remember, of course, your body has a different plan than you. We all know what tolerance is. Your body gets better at processing alcohol the more you expose it, right? So the more alcohol you drink, the better your body gets at getting rid of the effects of alcohol, right? And you develop tolerance and you have to drink more to get the same effect, right? So a lot of this research is based on the idea that we're measuring initial sensitivity, kind of baseline biological response, but it's confounded with, well, how much has this person been drinking and have they developed tolerance? Are they insensitive to the effects of alcohol because they drink every day, right? That gets complicated. One of the solutions to that, one of the reasons I believe those models is a whole series of studies, I've done some of them, that looked at the flushing genes in people of Northeast Asian ancestry. Has anyone known someone from China? It tends to be China, Korea, or Japan who have a strong flushing response. Sorry, is it there? I'm trying to figure out who asked. Yeah, so let me talk about that in a second. But let me talk about the flushing genes first. So about half of people in those populations have at least one allele of a gene that prevents them from breaking down acetaldehyde. So it does not affect their metabolism of alcohol. It affects their ability to break down acetaldehyde. If they're heterozygous, meaning they have one of these alleles, it means a good chunk of their enzymes are inactive. If they're homozygous, so they have two of those alleles, all of their enzymes of ALDH are inactive. And that major pathway cannot break down acetaldehyde, right? And remember, there are secondary pathways, right? But for those people, even a small amount of alcohol produces a lot of flushing, a lot of sickness. They're basically a naturally occurring, genetically based, and a simple genetic story, right? It's just a single gene. It has a huge effect on their response to alcohol. So to get back to your question, my friends were doing research on these genes because they thought, we're going to find that Native American groups that flush have the same genes, and this will also help us ethnically link those groups and migration patterns and things like that. It didn't work out. So there doesn't seem to be variants of these genes that exist in Native American populations. Good question. It's a complex story. I'm telling you a very simple version of it to advance this point. Yes? I don't know about that one. I don't know that. Yeah, you're not thinking of sickle cell anemia? No. I don't know. So there's a lot of research on this. I only know some of the highlights. One of the things, let me finish this point. So this is a naturally occurring high response group, right? That we know it's from their genes. It's not based on acquired tolerance or lack of acquired tolerance, right? And we know that it has a huge effect on their experience when they drink, and it has a huge effect on alcohol use disorder. People with even one of these alleles are at very decreased risk of developing an alcohol use disorder, right? People with two almost never develop an alcohol use disorder. I've read entire papers about someone that had two of those alleles and drank a lot, right? If you find someone like that, you write a paper about them because it's like, how did they possibly do this? They have to drink like small amounts of alcohol throughout the day because they literally can't drink one or two drinks in a kind of a regular fashion without getting very ill. And we no longer can give people with two of these alleles alcohol in the lab. It's considered unethical, right? So this allows us for inferences about kind of baseline sensitivity. It's a little different, right? It's kind of like knockout mice, right? It's not exactly natural, right? It's a little different than natural sensitivity or insensitivity because those might be through different mechanisms. But at least it's proof of concept that strong sensitivity to alcohol's effect does reduce risk for alcohol use disorder, right? It also allows us, this neat side benefit of this is it also allows us to make inferences about alcohol and acetaldehyde. Remember, I talked a little bit about that in the beginning. We can't really do this, but we know if you just have these alleles and you have none of the others that change alcohol metabolism, then you, at a given amount of drinking, you're exposed to more acetaldehyde than other people because your body's breaking it down more slowly. So then if having these alleles increase risk for a certain type of cancer, that cancer is more likely caused by acetaldehyde than alcohol, right? If people that have these alleles have the same risk of a certain type of birth defect, then we can make the inference that that birth defect is more likely caused by alcohol than acetaldehyde, right? Does that make sense to everybody? So most of the esophageal cancers that are caused by alcohol consumption, people with these alleles are at increased risk. We think some of those things are from the iatrogenic effects of acetaldehyde. Okay. All right, and then I was going to get to your point because there's variability in this in different ethnic groups because this is a complicated story. There are different alcohol metabolizing genes and different variants of those with different velocities. It gets very complicated. I wanted to tell the simple version of just the strongest one. Very briefly, alcohol affects judgment. This is one of those, yeah, I know that, right? People are bad at making decisions when they're drunk. They're also more impulsive when they're drunk, right? But there's lots of different variations of that, right? People do different dumb things when they're drunk. You know the person that just says stuff that they shouldn't say. And you know the person that does stuff that they shouldn't do. And you know the person that, so everyone's got a different profile of how alcohol impairs their judgment. And another thing that I do, I'll wrap up with this, right, is we look at perceptions of risk. So I'll just skip to the general thing, like the idea, let me skip over this. The idea that people make different judgments about what's a good idea, right? And it's a joke that people make about drinking, like, well, it seemed like a good idea at the time, right? Some of the behaviors that I'm interested in are driving related, and I don't really care what people think about drinking and driving when they're sober. That's a terrible idea, right? I care what they think about it 5 or 6 drinks in, because kind of axiomatically, they can't make the decision to drive impaired unless they're impaired, right? So when I did a study, the design of which is complicated, and I looked at, hey, how dangerous do you think it would be to drive, right? Given how impaired you are, right? Asking that same question when people were intoxicated, there was a huge effect of alcohol reducing their perceived impairment of driving, right? And it gets a little complicated, but the thing to me that's interesting is the yellow versus gray, that there was a big effect of later in the alcohol session, but those two groups are matched on BAC. So one's at 0.07 on the way up and one's at 0.07 on the way down. They have the same driving skill at those 2 points on the BAC curve, but they see driving as much less dangerous. And one of the things I want to talk about as the end thing is acute tolerance, which is recovery of impairment within a single drinking session. So if you know what regular kind of what we call chronic tolerance is. Acute tolerance is your body adapting to the presence of alcohol within a single drinking session. So your body's saying, well, clearly this chemical is going to be here and you keep taking more of it, so we're going to compensate for it, right? And if you get someone to 0.1 and keep them there for 4 hours, they're going to feel much less impaired at hour 4 than they feel at hour 1, right? Think about what that means, though, for people's drinking and judgment, right? People think they're less impaired, even when they are impaired, the longer they've been drinking. Most of the bad decisions that people make during a drinking session are based on the idea that they're feeling less impaired, they're feeling less drunk, and some of their actual impairment goes away, right? Reaction time starts to come back, again, even keeping BAC constant. But driving skill is not one of the things that comes back, right? And some of the other forms of impairment don't come back. And I think that I will wrap up there. This is the last thing I want to say. This is, in my lab, I asked people, based on how you feel right now, would you drive a car? This is at 0.07, so it's under the legal limit, but it's still a bad idea, just to be clear. The black line is people in the alcohol condition on the ascending and descending limb. On the ascending limb, so 0.07, right, about just under 20% of my sample said they would drive. On the descending limb, just under 60% of my sample said they would drive, right? That, to me, that huge effect is a lot of what I study, that as the drinking session goes on, they're at the same BAC. Their driving skill has not started to recover, but they're more likely to drive. And what the rest of my talk would have been is that I'm now doing this with smartphones and portable breathalyzers so that I'm actually measuring these things as they're occurring in the actual bar, because I want to know how people make the decision about whether they're too impaired to drive, at 1 o'clock in the morning, at the field house. So that, I'll wrap up there because we're out of time, but I'll take any questions. Thanks. I have a thank you slide, and these are some of my collaborators, and I should say that all my grants are funded, pretty much all of them are funded by NIAAA. I have no other conflicts, though. Yeah? Yeah, so 2 good questions. So I skipped over the kind of explaining the flushing response because I was starting to run out of time, but in general, when we talk about flushing, it's a buildup of acetaldehyde that people do get just from drinking a lot. If you drink a lot very quickly, your body will start metabolizing alcohol. You'll have a buildup of acetaldehyde. They get kind of red. This is when people start sweating. And sometimes that's a precursor to vomiting or other kind of acute alcohol poisoning things. The flushing response is from that buildup, and it's very pronounced. You can Google pictures of the Asian flushing response online. You could see when we used to be able to do research, laboratory research with people that had these alleles. They'd take a picture of them before they drink. They'd take a picture of them after 2 drinks, and they're just beet red, sweating a little bit, and they feel really strong negative effects of alcohol. Again, it lets us determine a little bit about how much of that is acetaldehyde, because that's what that gene does. No, not that kind of flushing, like flushing red, yeah. And then your other question is about sleep. So I always say, well, actually, there's a handful of people on campus now that are doing sleep and alcohol research. There's a good reason why we don't prescribe alcohol as a sleeping pill. It does not do a good job of helping people sleep. It does do a good job of helping people relax, but it actually disturbs sleep. It's a complicated process, but in general, I would say it reduces quality sleep, even if it improves the number of hours of sleep.
If you have any questions, please raise your hand and I'll bring you a microphone. But we're YouTube live streaming, so we want to be able to hear the questions.
I'm still trying to justify my own drinking. And so I'm thinking back on these epidemiological studies, and I'm thinking, I mean, certainly they measure illness in the abstention group and in the drinking group and can control for that and a lot of other demographic variables.
Yes. So
statistically.
Yeah, so that's a good question. I don't want to say that I have an answer to the question about whether the moderate benefits, the health benefits of moderate drinking exist, because I would say in the last couple years, it's been called into question a lot. But I would say about 4 years ago, I would say, hey, we haven't done the experiment, but all the research is pointing in that direction. There are tons of very good, well-designed epidemiologic studies that suggest that moderate alcohol use does have cardiovascular, at least, health benefits. It's just that some big studies in the last couple of years have questioned whether they've done a good job of what you're asking about. Have they done a good job of measuring kind of baseline differences in health? And the only way to be sure is an experiment, right? So that's kind of where we're at. Again, kind of going back to the theme of humans are terrible research subjects. I think the weight of evidence still leans a little bit toward health benefits of moderate drinking, but it's questionable at this time. Yeah.
I'm old enough that when I was pregnant, they recommended a glass of wine.
Yeah.
Now they're recommending that you don't drink. Can you
Absolutely.
explain that?
Yes. So I'll keep with the theme of humans are terrible research subjects. Think about what we have to do in order to understand how alcohol negatively affects pregnancy. You can't do that experiment, right? You can't randomly assign women to drink at different stages of pregnancy. So a lot of it is done through animal models. Some of the best fetal alcohol research in the world is done through different types of rodent models. There's some research where they'll go to naturally occurring, I know in Russia and parts of South Africa, there are naturally occurring high drinking groups of women that just don't believe this, and so they're going to drink this, and we do study that. I would say in the last 10 to 15 years is when we've really started to know that it's any drinking at all, even drinking in the first couple days of pregnancy, which is something that concerns me sometimes because those people might test negative on a pregnancy test, but when it's not a whole lot of cells, that is when alcohol could have the biggest effect. And so I think they used to also, my mother drank beer when she was nursing, right? Because they used to tell her, don't do that either. So I think just the quality of evidence improves over time, and we just didn't know anything about this. And really, a lot of alcohol research on humans didn't explode, I would say, until the 70s, right? So some of these questions just weren't really probed in the way that we would like. Other questions?
All right, if there are no other questions, let's have another round of applause for our speaker.
Thank you.